So they need attachment organs. Here I am just going to show you the cystos that prevalent in your country. Otherwise, they are too many. The Echinococcus granulosus, the larval stage, which is hydrophilic cyst, Tinea saginata, Hymenoleucus nana, okay? And also you don't have Tinea sodium because its importance, medical importance, because it is invading as a larval stage in different tissues, which is called cysticercosis. I'm going to compare the scholars and the gravid proglottid of Tinea sodium with Tinea saginata. Otherwise, the other parasites are prevalent in your countries and neighbor, in your country and neighbor countries. Okay, Tinea saginata is one of the most important prevalent tapeworms in the Middle East. It is called beef tapeworm because the intermediate house is cattle. As an adult stage, it is living in the intestine of human. In the scolex, they just have 4 suckers. They don't have any hooks. The size may be up to usually 4 to 6 meters. I had a samples from a lady referred to our lab. The size was this big. I couldn't believe it myself. Okay, just part of the strobila is prepared here to show you how big it can be. So it is part of the strobila or body of chain of the segments of Tinea saginata, okay? Human beef tapeworm. So they are living in the intestine. The last terminal proglottids are full of eggs. Gravid proglottids are full of eggs. Actually, just single gravid proglottids can have about 100,000 eggs. Can you imagine? 100,000 eggs, like this one. This is gravid proglottid of Tinea saginata, like this one. And all these dark spots of masses of eggs. This is the genital pole. In the gravid proglottids, we just have uterus and the eggs. In order to identify is it Tinea saginata or Tinea sodium, you need to consider the main stem of the uterus and count their branches. If the number of branches on one side is more than 13, usually between 20 to 23, it is called gravid proglottid of Tinea saginata, like this one, okay? But this is mature proglottid. In the mature proglottid, you can see both male and female reproductive system because all the tapeworms are hermaphrodite. It means every single tapeworm in the mature proglottid have both male and female reproductive system. Here, for example, this is the ovary, which is bilobed for Tinea saginata. This is the vitellaria, which help for the development of embryo. This is genital pole. Genital pole in the Tinea species is lateral and irregular. It means it may be in the right side, left side, and all these small dots, everyone is one testis, and they have vas efferent, vas deferent, and fertilization can happen just in one single proglottid or from one proglottid with another proglottid, or even in case of presence of many tapeworms like Echinococcus granulosus, actually cross-fertilization happens. After fertilization and production of the eggs, just uterine remains and all other part of the reproductive system will disappear because the tapeworm doesn't need them anymore because they have too many eggs. This is egg of Tinea species. Actually, all Tinea species and Echinococcus species, their eggs are morphologically identical. We just can identify them by molecular methods. So here I'm just writing Tinea, I don't know, is it Tinea saginata, solium, or even Echinococcus granulosus. Okay, inside the egg of all tapeworms, there is an embryo which is called oncosphere. In the oncosphere, you can see 1, 2, 3, 4, 6 hooklets. Okay? Do you know what is the role of hooklets? Hooklets means a small hook. What is the hook's function? Attachment and penetration. When the egg is eaten by intermediate host in the intestine, the other layer, which is called embryo fold, is released. But if the embryo wants to develop to the larval stage, they need to penetrate. So the hooklets helps the embryo penetrate the intestinal wall, and through the blood they move to different tissues, and in the muscles, striated muscles, they develop to larval stage. The larvae of Taenia saginata is called Cysticercus bovis. Here I want to show you the gravid proglottid of Taenia saginata. This is actually unstained, like this one. Unstained gravid proglottid that is noticed by the infected patients because they are moving to the anus. After staining in the laboratory, we can see and count the number of branches. Here, if you compare with the gravid actually proglottid of Taenia solium, if you consider this main stem of the uterus and count the number of branches, it is less than that of Taenia saginata. Here they are more than 15, but here they are less than 13. So in any part of the world, for example, China or Mexican, actual Latin American countries, that they have both Taenia saginata and Taenia solium, discrimination is important because Taenia solium is more dangerous because of what? Because of larval stage. For Taenia solium, the larvae invade the internal tissues, leads to cysticercosis of the eye, the brain, muscles, and so on. So it is very important, especially in the countries that pig husbandry is common. Right. So I told you about the morphology of the egg. You can see embryo. The outer layer of the Taenia egg is very dark, striated, and I say it is very similar to bicycle. Okay? Right. This is the larvae of Taenia saginata, which is called Cysticercus bovis. This is the muscles of a cattle heavily infected with Cysticercus bovis. The size is a few millimeters, and here you can see the scolex, like the adult stage, has just 4 suckers without any hooks. Actually, the life cycle, I can tell you here. We have the adult in human. Human is the only definitive host. Last terminal proglottids full of eggs are released in the environment. When the proglottid are eaten by the cattle during the grazing, every single embryo can develop to 1 Cysticercus bovis. Here you can see part of the muscles of the cattle, which is heavily infected with Cysticercus bovis. So Cysticercus bovis is the larval stage, so cattle is intermediate host. How human get infected?
Accidentally?
No, I mean by which food or actually other root. How human get infected with Taenia saginata? By eating such infected muscles. When the muscles of infected cattle is eaten as a raw or semi-cooked food, like what? Like kebab, steak, okay? The Cysticercus bovis develop to adult Taenia saginata. So the life cycle, adult in human, eggs and progloted, gravid progloted in the environment. They are eaten by the cattle. In the cattle, they develop two cysticercus bovis. When such infected meat is eaten by human, the larva develop to adult. But usually we just have one adult in human, but too many cysticercus in the cattle because of the small size. The life cycle of Taenia solium is very similar to that of Taenia saginata, except what? Except the intermediate host. In case of Taenia solium, definitive host is again human, but intermediate host is pig. When human digests the larvae from the infected muscles of the pig, they develop to adult. So it is prevalent in any countries they use pig. But one of the important aspects difference between Taenia saginata and Taenia solium infect human also to the larval stage. And it is very dangerous because the larvae, like cattle, muscles can develop in the muscles of human, in the subcutaneous tissues, in the eye, in the brain, and so on. So it is, in respect to the medical importance, is more dangerous compared to Taenia saginata. So for Taenia saginata and Taenia solium, you need to see the egg. You need to compare the cysticercus bovis and cysticercus cellulosique, which is the larva of Taenia solium, but the egg are the same. And you need to see actually adult Taenia saginata, gravid progloted, and also stem gravid progloted of Taenia saginata, Taenia solium compared, and to see the mature progloted of Taenia saginata compared, actually having both male and female reproductive system. Let's move to the next. So this is Taenia solium. In the scolex, you need to see the actually hoops. In addition of the suckers, they have hoops. The eggs are the same. I told you about difference of the gravid progloted of Taenia saginata and solium. This is the scolex. Both scolex of adult and larval stage has 2 rows of hoops in addition of suckers. This is cysticercus cellulosique in the muscles of a pig, very similar to this one. You see, they're exactly similar, except this is the muscles of cattle, but this one is the muscle of a pig. Right. The next taper is Hymenolopus nana. It is actually known as dwarf taper, because it is very small. I told you Taenia saginata is several meters in length, but Hymenolopus nana is just a few centimeters, and you can see all the taper in just one glass slide. In the scolex, like other cyclophilidian tapers, you can see 4 suckers, rostrum, and 1 row of hoops embedded on the rostrum. This is the scolex. In the actually gravid and also mature progloted, they are wide. They are wider than they are long. So they are trapezoid in shape. You see, they are very wide. Here is one. This is one. So they are very wide compared to the actually their long. Okay. Respect to Hymenolopus nana, you need to see the adult taper and the egg. Like other tapers, inside the egg you can see embryo, which has 6 hooklets, and the inner layer, and this is outer layer. The outer layer of Hymenolopus nana is thin. It's not striated, okay? This is difference between the egg of Hyena lepis nana and Tinea. And this parasite, as you know, is more prevalent in children. Echinococcus granulosus, one of the most important tapeworms. It has wide geographical areas. It is prevalent in actually Middle East countries. It is important, one of the most important tapeworms of human adults are living in the intestine of dog. They are just a few millimeters in length, so the number of proglottids is just a few, for example, 1, 2, 3, 4, 5, 3, 4, 5, okay? All the tapeworms can be found under the microscope because the size is very small. When the size of the tapeworms is small, so too many of the tapeworms can be invade the intestine of definitive host. This is part of the small intestine of a dog heavily infected with Echinococcus granulosus. Here you can see the scolex with suckers, hoops, immature, mature, and terminal gravid proglottids, and these are the eggs. The eggs are similar to the egg of Tinea species. And here in the lateral part, you can see genital cord. Okay, the eggs, as you see, are very similar to the egg of Tinea species, okay? When the eggs, actually the proglottid full of the eggs, or eggs are eaten by intermediate hosts, which are a variety of domestic and wild herbivores, and also humans. In the internal tissues, for example, liver, like here, the cyst develop. The cyst is called hydatid cyst. Here you can see liver with a mother cyst. Inside the mother cyst there are many daughter cysts. Daughter cysts are just smaller, but structurally they are similar to the mother cyst. It means they have laminated layer, germinal layer, broad capsules, protoscoleces, okay? If we prepare a section of the infected liver and stain, for example, it is hematoxylin and eosin staining, here you can see the normal part of the liver. This is the fibrotic tissues of the host. Here is laminated layer of the cyst, this thick part, which is acellular. This part, germinal layer, is darker. Why germinal layer is more dark? Because of the presence of nucleus, okay? This is germinal layer. The germinal layer produces broad capsule. This is broad capsule. Inside the broad capsule, there are too many actually protoscoleces. Every single protoscolex is capable of producing one adult Echinococcus granulosus. So when such infected liver is eaten by dog, all right, every single produce one adult Echinococcus granulosus. Here you can see the same picture with higher magnification. This is the laminated layer, this is germinal layer, this is the broad capsule, and these are the protoscolex. Protoscolex are also known as hydatid sand. Why? If, for example, this liver of sheep, the fluid is sedimented, at the sedimentation you can see many protoscolex, daughter cyst, broad capsule. So the other name for the especially protoscolex is hydatid sand. Hydatid sand is the same as protoscolex. Okay, here I think I showed you the Tinea. Here we have two samples from abattoir. This is part of the liver of the sheep which is infected with hydatid cyst. Actually, in the liver of human, we have the same scenario. When human digests the egg of Echinococcus species through infected actually water, vegetation, or direct contact with dog, the egg develops to a cyst which is mostly in the liver after liver in the line. So 90% in the liver and the line, and the other 10% to any other part of the body. Okay? So I expect you see all these macroscopical samples. You need to compare the egg of Taenia with the egg of Hymenopus species. You need to compare Cysticercus bovis with the Cysticercus cellulosae, okay? You need to compare the gravid proglotid of Taenia saginata with Taenia solium. You need to see the different part of the productive systems in the mature proglotid of Taenia saginata. Here we have adult Echinococcus granulosus and section of the cyst. You need to find different layers of the cyst as I mentioned to you. Okay, you can see, ask me your question, and before leaving the class, anyone who is interested can tell me about their identification on the samples. All right.